So, hello, I'm Hugo Bellin, and as the president of the Genetic Society of America, I'm excited to welcome you all to the first GSA Award Seminar Series. These online events give you a chance to learn more about the fantastic work that the 5-year Society Award recipients have achieved over the years, and I hope you will join us for the 4 other outstanding talks in this series, including Ting Wu's tomorrow at the same time, Ed Smith on May 5, Doug Koshland on May 11, and Feng Zhang on May 26. Today's speaker is Ruth Lehmann, who's a recipient of the 2021 Thomas Hunt Morgan Medal for lifetime contribution to the field of genetics. And the Morgan Medal recognizes the full body of work of an exceptional geneticist, along with a strong history as a mentor. Ruth received her medal last month in a ceremony at the annual Drosophila Research Conference, where she was recognized for her groundbreaking work on the unique biology of germ cells. Focusing on the only cells of the body that are able to generate a new organism, the Lehmann Lab made a series of highly influential discoveries that illuminate many aspects of animal development, including cell migration, cell signaling, RNA regulation, genome integrity, and inheritance. So let's welcome Ruth, and without further ado, Ruth, it's over to you. You can share your screen.
Okay, thank you so much. I'm going to share my screen now. That's right. Okay, you see just one slide, or do you see?
No, we're in presentation mode.
Okay, that's why we're testing this. Okay, so it's a real pleasure, and I am so excited about receiving the Thomas Hunt Morgan Medal for a fly geneticist. That's sort of the biggest achievement one can kind of think, wonder and wish for, and I'm so thrilled. And I looked for some good picture of Morgan. I love this one in the fly room. You know, I love the suit and then the mess of the fly bottles. And apparently it was a real mess in that fly room, and but you know, which led to discoveries of what phenotypes mean, how you correlate genotype to phenotype, and genes being on chromosomes and recombination, and so many tools. And so obviously he is the founder of our field, but for me there is another really and personally most important person, and that's Christiane Nusslein-Vollhardt. I did my PhD in her lab, and she really told me how to do bold science, and she really, by her and together with Eric Wieschaus initially, and then in Tübingen, the work in her lab on pattern formation and systematic genetic analysis of flies really led the way to where I and my lab, who I will acknowledge later, are today. And so it started in Christiane Nusslein-Vollhardt's lab working on pattern formation, but what I want to tell you about today is more sort of the work that followed up from that in my own lab. And whoops, I think we jumped. Oops, things are not going my way here. Okay, here we go. So germ cells are forever, and I want to focus on germ cells today. And as you can see here, obviously genetic information from father and mother is passed on to the next generation, and on the way it's actually mixed up. That is obviously what is the underlying part of genetics and what we study. However, absolutely critical to the transfer of information from generation to generation are the germ cells. And so germ cells are specified in the early embryo. They then migrate to the somatic gonad. They then are important that their genome the integrity is preserved when they're going through meiosis, where voluntarily double-stranded DNA breaks are made to mix the genomes of the mother and the father to generate then egg and sperm respectively, and the cycle can go on. And so together with the genome integrity, which preserves the genome as healthy as possible, of course, the germ cells are also the drivers of evolution. And it is an incredible dichotomy in a way between these two conflicts that makes studying germ cells so spectacularly exciting. But there's something else that in particular my lab is focusing on and has focused on, and that is that there is a big discrepancy between the contributions, the relative contributions of male and female. The human egg is about 1,000 times larger in volume than the human sperm head. This is very similar for the fly egg. And it is really the contributions that come from the egg that far exceed the contributions which are purely coming from the two nuclei, which eventually will give the genetic information to the embryo. So the egg provides additional information. And that's very clear when we think about mitochondria, which are obviously incredibly important for the development of the embryo, for the functioning of pretty much every tissue, and they are only passed on through the female. And another aspect of, and that's particularly important, beginning with in the fly embryo already, are germ granules, which are already found in the early oocyte, when the oocyte when it is maturing, and then these germ granules will be there throughout the lifetime of the germline development, and they are carrying critical aspects for germ cell fate with them. And so these granules are really the hallmarks of all germ cells. So I was just telling you in flies, they're already in the embryo transmitted from the mother, but actually irrespective of how germ cells are specified in the embryo, germ granules, these dense membraneless particles, are found in all germ cells and throughout development. So for example, as the Barbiani body in oocytes, here we have examples of embryonic germ granules in C. elegans, Drosophila, and zebrafish. And then there's also the nuage, where these granular components are associating with the nucleus, and that can be in the differentiating egg or in the differentiating sperm. So we are very interested in what these granules are and how do they fulfill the particular functions which are so germline specific. And it seems particularly important because these germ granules are involved in RNA regulation. And when we initially identified genes which were important for germ cell formation and germ cell function in Drosophila, we were stunned in a way to initially realize that none of the proteins we identified had simple homologs. But then when the homologs were identified, it became clear that these homologs, which are specific for germ cell fate, seem to be mostly encoding for proteins which are RNA binding proteins and have a role in RNA regulation. So that's very different for other fates, like somatic fates, where transcriptional master regulators are the defining and also in most cases very conserved components which tell us about fate. So somehow the germ cells are regulated very differently and their fate is determined and maintained very differently from somatic cells. And so in flies, as I said, and I hope, can you see this pointer? In flies, the germ cells are set aside in this, or initially these germ granules are formed at the are found at the posterior pole, and you can see here in more details the individual granules within this germplasm region, and then you can see an electron micrograph here, a structure of a germ granule. And I did see Tony Mahowald being on this Zoom, and so I want to point out that Tony has done an enormous amount of work to tell us about these granules. And so what we have found more recently is that the way these granules, they are membranous granules, and how they form is really that they form RNA protein condensates, and that this is done through demixing from a solution. And so that we get enormous concentration in the germ granules of proteins, while there is hardly anything in the neighboring intergranular space, which is very similar to the concentration in the soma. And so this and many other experiments that we and others have carried out suggest that these granules form by a process which is called phase transition, which is sort of more like how oil and vinegar separate in your vinaigrette. However, that said, the fly germ granules are not liquid. They have actually quite a bit of organization, but this is how they assemble initially. And so we know quite a lot about how these granules assemble, and we know that the components are really all RNA regulatory proteins and the major components, and there are germ granule specific components, and then there are also other components which are shared actually with other RNP condensates like stress granules or P granules, P bodies. And Oscar is the scaffold protein for the germ granules. That means wherever Oscar is in the embryo, that's where these granules will form. And then it recruits these other components, which are really important. And while Oscar is not really broadly conserved, these other components are highly conserved, Vasa, Tudor, and the Argonaut protein family. And they are conserved for the germline granules, for the particular germline granules. What we found early on was that the RNA of some of the granule components here, in particular nanos, is highly concentrated at the posterior pole, but you can see also RNA throughout. And we and others then identified more than 200 effector RNAs, some of which actually only a small percentage is similarly highly enriched at the posterior pole. What seems to be the case for quite a few of these localized RNAs is that they are translated specifically at the posterior pole, and we think they're actually translated within the granules. And these protein products that are then produced are important for germ cells formation, so making them actually in the first place, specifying them, and also for the migration of germ cells later to the somatic gonad. The RNAs, which are then sequestered in the germ cells, are prevented from degradation initially. They do degrade eventually. But in addition to mRNAs, which are sequestered in the germ cells, are also piRNAs. And these piRNAs, maternally provided piRNAs, because all of this is independent of zygotic transcription, these maternally provided piRNAs provide already a protection of the germ cells against transposable element activities. And so there's the transposable element defense, which is also directly associated with germ cell formation. And we wanted to understand the germ plasm and the germ granules in a more quantitative way. The problem was, since the RNA is everywhere as well, grinding the embryo up will give us sort of a clue of everything that's in the embryo, but will not provide insight into specifically what's actually happening at the posterior pole. And so when Tatjana Trek joined my lab as a postdoc, she has now moved on to her own faculty position at Johns Hopkins. She embarked on a quantitative analysis of what it actually means for these RNAs to be localized or enriched at the posterior pole in the germplasm. And she used single molecule FISH to be actually able to count single RNA molecules of, in this case, nanos RNA. And you can see these single dots here, and then extrapolate from these larger how many RNA molecules are in there. And so with this quantitative analysis, she was able to establish the quantity of the RNAs in general in the entire embryo, and then see how only 3 to 4% of the total RNA localized is highly enriched in this about 0.1% of the egg volume. And this is at least an enrichment of 8 to 10-fold. And so that kind of is the same idea or concept that I was telling you about before about these biocondensates, because now we have a situation here where the concentration of these RNAs is a lot higher than everywhere else, and having the RNA so concentrated, having the proteins which make up the granule so concentrated, will allow biochemical interactions to occur that normally could not occur in the diluted state. But all of this happens within the same cell, and that's the fascinating part. How is this organized within one cell, the big egg cell? And so what Tatjana did is she wanted to understand better how these RNAs and proteins are actually organized. And for this, she used an algorithm that was developed by Timothy Lionhead, and we also collaborated with Harry Schroff to use their structural illumination microscope to see how the RNAs related to each other and how the RNAs in the germplasm related to the proteins and how the proteins related to each other. And since everything is so dense, colocalization per se, just yellow spots, don't tell us so much. And so this algorithm measures actually the distance between the peaks of fluorescence and then gives a ranking to how consistent that distance is. So if two molecules are really highly co-localized, then they should always have about the same distance in the peak of their fluorescences. If they're, on the other hand, highly concentrated, but they are not in a relationship with each other, that distance should change, and that would give us a smaller R value, so going from 1 to 0. And so if you look at RNA, if you look at proteins like OSCA and VASA, which we actually know they're interacting, we know this by pull downs, we know this by structural biology, they're clearly co-localizing. But the surprise was at the time that it seems that the RNAs are really differently organized within the granules. Some of them seem to be quite nicely co-localized with the proteins, but some don't. And so when Tatjana analyzed this then more concretely, measuring actually the distance of the RNA from the center of VASA, highest VASA fluorescence, which would be the center of the granule, we are assuming. We have a nice control here with OSCA protein. It's highly co-localized. When we label an RNA with 2 fluorophores, but it's the same RNA, we can gauge what the optimal co-localization is. But we also realize that some RNAs are more in the center of the granules, while other RNAs are more at a distance. And what we realized is that the RNAs of the same species seem to be forming these homotypic clusters within the granules. And we can also show this by basically establishing the same map by looking at how the RNAs correlate to each other and what relationship they have, this showed us that within the granule, we have actually an organization of the RNA. So the granule is much more organized than everything just coming together in one blob. We found that surprisingly, the organization of the RNA, so where the RNA is within the cluster and how many RNA molecules there are, is not correlated with the amount of protein. So this tells us that there isn't a simple one-to-one relationship where the protein actually determines the organization of the RNA. It seemed like the organization of the RNA was really much more self-organized. And so there are sort of two ideas of how RNAs could be organized. A really favored idea that we had came from the work mostly from Anne Frussi's lab, which really told us how the OSCA RNA is transported. And what she and her lab found was that there is a little stem loop in the 3' UTR, and this stem loop can actually kiss with a stem loop from another OSCA molecule, and that is important for RNA transport. And similarly, kissing loops had been shown for bicoid RNA-RNA interaction. But there are other models, and these other models have really come from the analysis, like an example here from the Parker lab, where RNA aggregates are forming, and they're more dynamic than requiring specific sequences, which of course with complementary mutants can be really illuminated. So these are much more dynamic interactions, which are more on and off states. And so we wanted to know how these homotypic clusters are forming. And so here's again the example of OSCA, where we have this dimerization loop, and that allows the localization of OSCA. And we have these palindromic sequences here. And so what we would expect if it was like in OSCA RNA, where two RNA molecules can be transported together, if we have a heterologous piece of RNA stuck to the stem loop, then that RNA should be equally able to be co-transported and should be co-localized. So we would expect that co-localization of the same molecule plus co-localization of a similar molecule carrying the same loop should have the same, should co-localize similarly. And so that would be the idea, irrespective of what's hanging on to the RNA. It's the 3' UTR and specific sequences in the 3' UTR. So we set out to look at this kind of analysis, and we know for nanos also that the 3' UTR is absolutely important to get nanos RNA into the germplasm. But there were some surprises. So why the correlation of course, the same RNA again is really high, which is what we would expect, labeling the same RNA with two different fluorophores. But if we use the same 3' UTR, same 5' UTR, same promoter, but we have two different protein coding sequences, we don't see co-localization. They organize into homotypic clusters in the germ granules, but they're not co-localizing. And even more extreme, if we had actually exactly the same nanos molecule, all the pieces of nanos, but they just were distinguished by two heterologous sequences, they did no longer show co-localization. What that tells us is that there is clearly a sequence specificity, which means the sequence, the genes are recognizing each other, the transcripts, the same transcripts can recognize, but we cannot identify a specific sequence which is required for this kind of organization. And so this is not 3' UTR dependent, despite the fact the initial accumulation and enrichment in the germplasm is, and it is sequenced apparently not dependent on a specific sequence. So this was really puzzling. And so we wanted to have another completely different part of kind of evidence for this. And so we thought about what does it actually mean when two molecules are tightly and sort of closely associated. What it would mean is that at some point those sequences in trans should be in a smaller orbit because of their close association, while other sequences further away, and so in this case the 3' UTR, for example, the stem loop in Oscar, should be closely associated, while other sequences of the RNA should be sort of further away, and so this would be kind of like a rosette or a bouquet. While if the interactions are completely dynamic and they can be with any kind of the RNA, it is just a property of the RNA, we would not expect to find such an association. And so we used STORM, stochastic optical reconstruction microscopy, to reconstruct the dynamics of RNAs following them along their sequence. And so again, we know lots about Oscar, and so that was really our positive control. If we could actually map by just imaging this dimerization domain, and it was pretty amazing. So using different fluorophores probes set along the entire sequence of the Oscar gene, we found that when we probed for the orbits in which we see the fluorescence for the regions which are around this stem loop, then it is in a much smaller orbit than the other. So really confirming the kind of rosette structure. And indeed, we haven't tried this, but one could think that one can use actually this technology to map these kinds of interaction domains in RNA. So this is kind of the first example of in vivo mapping actually an RNA-RNA interaction domain. But when we did the same experiment in nanos, despite the fact nanos has some beautiful stem loops that we were very excited about because they had the same kind of palindromic sequence, all that, but in genetic tests, as well as in the STORM analysis, they did not show to be specific interactions. Instead, the RNA-RNA interactions are dynamic. And so what we conclude from that is that there are two different ways which we can distinguish with the two different types of experiments that I showed you, the genetic analysis of mapping sites in the RNA, as well as the image analysis with STORM, that demonstrate specific interactions. And then this organization, which is a dynamic global RNA interaction, and so the global mRNA provides really the specificity, while and the mRNA binding proteins, as I showed you, are permissive for this. And we think that this reflects a disorganized intermolecular interaction that is more dynamic and may actually play an important role when we're thinking about how these RNAs are regulated. And so now I want to come to another part of the story and briefly and tell you a little bit about mitochondrial inheritance in Drosophila. And so what's quite interesting is the germline. We have these germ granules, and actually mitochondria are often associated with the granules. And here you can see a particular nice tomogram which shows us this association between the granules and mitochondria in the germplasm region. And this association is very common for oocytes. So whether this is a mouse oocyte, and we have the intermitochondrial cement, they have the mitochondrial cloud in Xenopus, or the germplasm in flies. Mitochondria are only inherited through the female germline. And mitochondria have their own DNA, and this DNA can mutate 10 times faster than the nuclear genome. So there is, and there's little or no recombination. And so this RNA, this mitochondrial DNA is very vulnerable to mutations. And the variability between mitochondrial DNA molecules creates a heterogeneity, and this heterogeneity can affect the penetrance of mitochondria. mitochondrial diseases. And so these germline mitochondrial DNA mutations are a major cause of inherited metabolic disease. But what this mutation rate and the lack of recombination also tells us is that there is, without selection, there is something, and here I have to bring up another wonderful fly geneticist, Muller, and this is the Muller's ratchet. What Muller predicted is if you didn't have recombination and you accumulated mutation, you basically would die out. So something has to happen to select for mitochondrial DNA molecules, mitochondria, which are functional. And so there are two aspects to this. One is a purifying selection, where mitochondria which are mutant will be depleted, and the other is a bottleneck, where as the mitochondria are subdivided into the germ cells, some could be taking on pretty much mostly good functional mitochondria and some non-functional mitochondria. And Thomas Hurd, together with a former postdoc who's now at the University of Toronto, and Toby Lieber, who is a senior scientist in my lab, set out to test this idea of selection. And what they used was a heteroplasm where we transplanted wild-type mitochondria, which wild-type mitochondrial DNA, into a mutant mitochondrial background. And so most of the mitochondria are actually mutant, and only a small percentage is wild-type in this setting. And the important part is that this mutant has a temperature-sensitive mutation, and so at the and I'll get back to that in a moment. We can distinguish the wild type and the mutant because we're actually using different species. We're using Drosophila jacoba, which we can probe with a specific probe, the mitochondrial DNA, and Drosophila melanogaster then with a melanogaster probe, and that probe works to wild type melanogaster as well as to this mutant. So what is this mutant? This mutant is a temperature-sensitive allele, which was identified in Pat O'Farrell's lab. And this can be selected against because it carries a mutation in the cytochrome C oxidase subunit 1. And at 29 degrees, we lose the predominance of this now non-functional species over generations. And so we wanted to directly image when selection is occurring. Pat O'Farrell's lab had clearly shown that selection does occur, and this has also been shown in many other species, including in mice and in humans. But we wanted to see when does it occur so we could actually probe for the mechanism. And so at the permissive temperature, the heteroplasm, which is wild type, is jacoba, mutant is Drosophila melanogaster. We see mostly Drosophila melanogaster. However, when we increase the temperature, when we grow these flies at the restrictive temperature, you can see how the selection is occurring. It is occurring in the germline. What you see here outside in pink is the soma. So the selection is only occurring in the germline. And now we could go back and ask when is the selection occurs. And we found that the selection is actually occurring not in the stem cells, but shortly after, when the cyst is developing. And what we found was that from this, going from the stem cells to the cyst, the mitochondria fragmented, so that in these fragmented mitochondria, there are fewer mitochondrial DNA molecules. So why a mutation could be compensated by functioning mitochondrial DNA. In this case, the mutation may no longer be compensated because now each mitochondrium has on average only one mitochondrial DNA molecule. So we wanted to test whether fragmentation is indeed the reason for selection, the starting point of the selection. And so we used mutations in both in mitofusin, which is required for the fusion, and similar experiments in DRP1, which is required for fission, to ask whether indeed this fragmentation is necessary and sufficient for selection. And what we found is if we don't have fragmentation, we see no selection. And if we have fragmentation in stem cells already, we can see that fragmentation is sufficient to see selection. Indeed, we could also knock down mitofusin in the soma, and now the soma was also undergoing selection. This suggests that the entire machinery for the selection process is not germline specific. It is just the expression of mitofusin, which is downregulated exactly prior to the selection step. So the model then is that we have these large mitochondria, which have different mutant and wild type DNA. Then we have a fragmentation step, and that leads us then to selection. Through many different genetic experiments, which are at the moment also ongoing, we are starting to identify the components and what is required. So we know that the necessary and sufficient step for this selection in the germline is the downregulation of mitofusin from the stem cell to the cis stage. We then know that there is a developmentally regulated known pathway, the NIX-BNIP3 pathway, which leads to mitophagy, and that this mitophagy may be distinguishing between mitochondria which can produce high or lower levels of ATP. And we also have evidence that there is also some aspects of selective replication. Interestingly, in former experiments, the experiments when we were looking at in general how the cysts develop, we found that during these stages, the requirement for ATP is actually low for differentiation. So it is not required for the development of the cysts at that stage. So this seems like kind of a clever way of just measuring ATP without making that measurement of ATP and the number of functional mitochondria at that time critical for the development of the and differentiation of the germ egg chamber. And so just to summarize, mitochondrial selection occurs in the female germline, in the fly during these early stages of differentiation. It is due to a regulated mitochondrial fragmentation. The mitochondria are eliminated by this regulated mitophagy. And this mechanism of selection per se is not germline specific. It is just the downregulation of mitofusin that makes it germline specific. And so with that, I just want to kind of complete and say how fascinating the germline is. Obviously, for me, it is. It's been an amazing journey on many different aspects. We are still very much excited and interested in how these granules are put together because they are no simple structures. They are clearly organized, as I could show you with the formation of homotypic RNAs. They're clearly important for RNA regulation, and they seem to be really essential in a way, the essence of germline regulation and germline fate. We are also interested in these aspects of symbiosis, like obviously mitochondria have to be transmitted, but there are other creatures which are transmitted through the germline, like the bacterium Wolbachia, which has a lot of positive effects. And so we are kind of struck by the idea that Wolbachia is sort of on its way to becoming a symbiont, just like perhaps at the beginning of when mitochondria entered the eukaryotic cell. And so I want to thank my lab, past and present. They are an amazing group. None of what I talked about would have been possible without them. And it's just so phenomenal. It's also wonderful to see them going all over. At the moment, this was actually our last get together in New York before the group split into a Boston and a New York group. And I also want to thank the two institutions where most of this work was, or all of this work was carried out. The Germ Institute at NYU Langone, which, and I was supported there by HHMI and the NICHD and other foundations. And now, as of late last year, half of my lab has moved to the Whitehead Institute. And we are very grateful also to all the support we have received, especially the Drosophila Stock Center, and also help from the Zhu and O'Farrell lab for the mitochondrial work. A lot of work from the NYU microscopy core for a lot of the imaging that I showed you, and our collaborators on the RNA localization project. And I thank you all for listening. And again, I'm so grateful to the GSA for this amazing award.
Thank you. Silent applause. All right. Does anybody have any questions? You can post your questions on chat. I don't see anybody who's posted a question, but I'll start with one. I'm particularly interested in the mitochondrial work and the role of other proteins that play a role in mitochondrial fusion and fission. And so you mentioned DRP1 originally. If you overexpress DRP1, do you see a phenotype or not, or a loss of mitofusin in the germline?
So DRP1 has basically the opposite effect. So consistent with the idea. So,
so both
it's
are
not regulated. So we're focusing on mitofusin because mitofusin is actually specifically downregulated. So that's the developmental, because it's kind of the interesting part is this is just like the mitophagy, right? It's not a disease caused effect. It's really, this is developmentally regulated. So we're kind of interested in seeing what is actually changing during those stages. And so from what we've seen is that it is mitofusin, which in this case is the regulatory step, but we can achieve the same, I mean, in opposite ways with DRP1.
We looked at mitofusin expression, and I'm surprised to see that it's not expressed in all cells.
No, it's really upregulated, quite remarkable.
In the germ
just
line.
at that stage, just, the stem cells are not, and then it's really very nicely upregulated, and it's very nicely downregulated at just after the stem cells differentiate, and that's kind of and it's sort of an interesting transition at that stage. So it's on the way to obviously when the germ cells are on their way to differentiation. So it's a real clear blip, and we have seen this in antibody staining. We have even seen it in RNA expression.
So there's a few questions, and so maybe we can start. The first question is, how conserved is the granule posterior localization across Drosophila species like simulants and others?
Can you say that again?
So how conserved is the granule localization at the posterior end among the different Drosophila species?
We have the expert here, Tony, who has done all the work. So this is not work that we've done, although we're starting to become more interested in looking at different species, obviously with the mitochondrial transplantations or mitochondrial DNA. But in Drosophila, you have germ granules at the posterior pole. In Drosophila species, that's what Tony showed. Tony showed also very nicely that the structure of those granules can be quite different, and that's very interesting. And then Paul McDonald had done a really nice experiment where he actually, so Oscar is this scaffold, is the seed protein for these granules, and so there seems to be some link between this scaffolding by Oscar and how the shape of the granules actually look. And so that's it's a very interesting. But what's also interesting, Oscar is, first of all, it's not very conserved, while the other components are all highly conserved. So Vasa, Tudor, and aubergine or argonoid components are in all germ granules in all species. And later in Drosophila germline development, you also have those three again. And in those cases, we do not understand as well what are the seeds. So we know the seed very well, and it's very nice to analyze it. But clearly how other germ granules are actually initially being seeded is quite different. So for example, in C. elegans, there's a very different way of how they're seeded, but eventually Vasa proteins are components of the germ granules also in C. elegans.
So the next question is from a non-Drosophilus, I assume, and that is, what are the consequences when germ granules do not form and function properly?
Yeah. So
Grandchildness.
yeah, so there are many points. So if you don't have any germ granules, then you can't make germ cells. And but also in Drosophila, we also know that nanos, the RNA, the protein or the gene that got me into much of this, nanos and Oscar. So Oscar is required for making the germ granules, but nanos is one of the critical RNAs. It also affects front to back polarity. So you have polarity effects in the embryo. We also know, for example, there's an RNA localized to the germ granules, which is Wunnen, and Maya Sladina in my lab showed that the amount of Wunnen dependent on how much Wunnen you actually get in the primordial germ cell will predict how well the germ cell will make its migration to the gonad, whether it will die or whether it will actually be able to continue the migration to the gonad. Similarly, there is a GPCR TRE1, which is also required for germ cell migration. And then there's also a peptide PGC, which is expressed in the germ, in this germ plasm, and is dependent on the granules to be translated. And that's important for transcriptional silencing, which totally turns down somatic patterning activity in the germ cells. And similarly, the germ cellus protein, which is required for germ cell formation, also turns down the somatic protein signaling pathways by degradation. So a really important function of these granules is to localize RNAs. And when those RNAs are translated, they actually repress the somatic fate.
All right. Question from Tom Klein. There is the idea that processes controlled by RNPs may reflect evolutionarily ancient processes. You mentioned lots of RNA binding proteins, but is there any evidence for the RNP complexes that require both an RNA component and a protein component to function in germ cell formation and/or function?
I'm not sure I'm understanding the question. So I would first say, absolutely, you know, everybody from the RNA world would say, you know, that RNPs are so important for germ cells because you could even argue the germ cells, they are the first cells, right? The first cell is sort of a germ cell. When you think of a yeast, yeast is a germ cell kind of. And so that it doesn't have a transcription factor, which makes it different, is perhaps obvious, right? And so, so in that regard, the RNA regulation part makes a lot of sense.
Ruth,
all of these RNA binding proteins are very conserved, so they have been there for a very, very long time.
Ruth, I'm thinking of complexes where you need the RNA component in order for the protein complex to do its job, absolutely. Like a ribosome, RNPase.
Now I understand your question. Yes, absolutely. So so in most cases for these condensates, and this has actually been studied much more clearly in vitro in C. elegans by Tony Hyman, also Geraldine Seydoux, very beautiful work, which has shown that to make these condensates, you need RNA. The RNA in those cases is not necessarily specific. So we cannot really take all the RNAs away, which we have, and we can actually, our granules seem to, while we can recapitulate some of it in tissue culture, and we did this a couple of years, we're not able to really recapitulate a granule that satisfies us to fulfill all the hallmarks of a germ granule. But it is very clear that for the condensation, RNA binding is actually really important. It's actually one of the valence. So when you think of the Oscar protein, which despite the fact it's kind of a novel protein, it is the epitome of one of those proteins which now we know is important for phase transition. And that is, it has an RNA binding domain, which they pretty much all have. It has then these IDRs, these unstructured domains, and then it has a protein-protein interaction domain, which in case of Oscar, the Lotus domain is both making dimers actually of Oscar, and then Oscar also interacting with Vasa. And that really helps to make the condensation. So if that protein now is at a higher concentration, and we have shown this in tissue culture, if we express it at a higher concentration, it will form a granule, and it will then recruit, for example, Vasa to that granule. And that is as a phase transition. But for that, you do need the RNA binding domain. So all the mutations in Oscar that I identified as a graduate student, they're all in the point mutations, they're all in the RNA binding domain.
So there's too many questions, so we're going to have to kind of do some selection. One question is about the messenger RNAs that are at the pole. What triggers their translation, and what are the spatiotemporal cues for these?
So I will give that to Roy Chen in my lab, who just gave his group meeting this morning, and that's what we're trying to figure out. But the interesting part is, because I'm seeing some C. elegans questions here. So the C. elegans germ granules are, as I understand it, a little different from the Drosophila germ granules in the embryo. And that is that the C. elegans granules are really their major function seems to be to preserve RNAs, to make sure that they're taken into the germ cells, and they're more in terms of a translational repression. The Drosophila germ granules, the RNAs are actually translationally repressed outside and then are translated in the granules. So that's sort of a different, and that's what makes these Drosophila granules very interesting, of what allows the RNAs there to be translated.
If germ granules are used to enrich germline specific messenger RNA, the PGCs, are there opposing mechanisms in the soma that enrich or deplete somatic messenger RNAs that are maternally deposited?
That deplete somatic. So the mechanism in the general mechanism which is happening in the soma is the maternal to zygotic transition, where really pretty much all of the or many of the components which were deposited in the egg are destroyed. The RNAs are destroyed, and so those RNAs are protected in the germ granules. I mean, they're not just protected; they're also enriched, but that mechanism is occurring in the soma. So there is an anti-mother process going on in the soma, which gets rid of most of the germline derived components from the egg. Some of them obviously are passed on. A lot of all the ribosomes, the Golgi, like the mitochondria, they're all given from the mother to the egg, and they are maintained. They're really important for embryogenesis. But there is a transition. So just like the germ cells get rid of don't have zymatic transcription, get rid of some signaling proteins which could provide somatic information, the soma is basically depleting a lot of what it got maternally, the maternal endowment.
So to come back to the C. elegans question, do you think there is a different kind of germ granules in Drosophila versus C. elegans with Z granules and mutator foci?
Yeah, so I do think that so there's a couple of differences. So for example, in C. elegans, if I understand it correctly, and I don't want to, but there are a lot of mutations. So first of all, because of redundancy, a lot of the genetics was actually very difficult to figure out which ones of the proteins are actually the sort of are the seeds. But it seems like I think from Geraldine's work that it's the MEG proteins. In many cases, you make mutations in these, you still get germline, because there's another mechanism which is really important in germ cell specification and which is kind of separate from the granules, as far as I understand, and that's the function of Pi1, which is a transcriptional repressor and represses the transcription. So both of these, the granule components and Pi1, have to work together. Also, the analysis of the liquidity, although there is also more structure, as Geraldine's work has shown, to the granules, that may be more extreme in Drosophila. So I think by comparing and contrasting, we're going to learn so much more. It is very interesting to think about the nuage, which is much more similar in all of these species, whether they're, whether we're going to get actually a complete grammar at some point of what germ granules are and how we coordinate their function, because the nuage is the site where the piRNAs do their job and the Argonaute proteins to generate actually the response against transposable elements. So that is another very conserved aspect, and that's like but whether we can learn actually whether there will be some besides the initially it's phase condensation, high enriched proteins and RNAs, whether we can figure out if there's some more structural features of these granules that are conserved and that are specific for germ granules. I think that will be very interesting, and we just need to get even better with our imaging and with some of the structural biology on those processes.
Okay, we're going to go for one last question, and that's another comparison with C. elegans and flies. So, losing certain components of P granules in C. elegans sometimes leads to phenotypes many generations later, like sterility. Do you observe any defects in Drosophila that accumulate over many generations?
So that's what I was saying. So in C. elegans, you have this slow process, and because so in
the question is it happening in Drosophila for someone?
No, it doesn't happen, because they either make germ cells or they don't make germ cells. At least that's what we know so far. So
it's black and white versus
pretty black and white, but it may be the genetics. So I wouldn't want to say it's absolutely different. It's just that either you have OSCA, you don't have OSCA. If you take OSCA and you put it to the anterior pole, you make germ cells there and they're functional. And so it's kind of, it's a little bit more black and white. So
there's no mutation in a partial loss of function mutation in any of the RNAs from the germ cell plasm that would lead to a systematic reduced, a grandchildless phenotype that is partial.
So yes, you can get partial. So we have OSCA alleles which are partial. We even halving the amount, but then it depends on what your genotype is in the next generation. As far as I know, it's not
inherited. All right.
Not inherited.
I think
also one difference, a large difference, sort of talking about biology, is that in flies, and as in all other species pretty much, the germ cells have to migrate. And then when only a single germ cell gets to the gonad, it can actually make a full-fledged ovary. We've shown this. In C. elegans, the germ cells don't have to migrate. Actually, the somatic gonad migrates kind of to the germ cells. And so that is one of the kind of, you could say, selection processes which occurs. And so, so when the germ cells are not totally 100% really good functioning, they usually don't make it to the gonad. So that's kind of what we have found. So I think that may be another quality part. And so I don't know. But I don't want to make it too, oh, this never happens. Maybe we have our genes.
Thank you for a wonderful talk and a very deserved award. And we're going to have to end here. The hour is over. And again, thank you, Ruth.
Thank you. Thanks everyone for
beautiful
listening.
seminar.
It was
Thank you.
really fun. Thank you so much. Thank you. Bye. Bye everyone. Bye all these friends and former trainees. And it's wonderful to see you all.
That's great.